Good morning, everyone. Greetings. Welcome to our next our apartment seminar series. It's a pleasure again to welcome one of our own, Dr. Phil Hahn. I did not realize that Phil was a triple badger. So bachelor's, master's, and PhD at the University of Wisconsin before he moved to his postdoc at University of Montana. He arrived in UF in our department in 2020 with a 50-50 teaching research appointment in infectious ecology. And we're going to hear all about this today, but Phil's work kind of focuses on the interface between population, community, and evolutionary ecology. I also want to highlight just the balance, like 50-50 split, we don't have that many, but Phil has a amazing balance, and that was on show last year. He received both the CAL's Early Career Excellence in Teaching Award and the Richard Jones Outstanding New Faculty Research Award. They're kind of like both the Early Career Research and Early Career Teaching Awards for the CAL, so kudos to Phil for balancing all that. He also received last year the Florida Entomology Society Annual Achievement in Teaching Award. Phil teaches a number of really important classes for our department: insect ecology, linear models, and agri-natural resources, and insect community ecology. And he also enjoys, while he's not receiving awards for his research, biking, hiking, and soon to be banjoing.
Okay, thanks everyone for coming. Thanks for that introduction. And as Andrew mentioned, he did a nice job summarizing my research program. So my program focuses very broadly on the ecology and evolution of plant herbivore interactions. And most of our work does take place right at the intersection of population, community, and evolutionary ecology. So we have a couple of themes in the lab. One of our major themes is evolutionary ecology of plant defense against herbivory. So we ask these questions typically across very large gradients. So this is work from one of my students, Jake Kirchberger, who's working on horse nettle or Solanum carolinensis across the whole distribution, the whole north to south distribution of the species, trying to understand how plant defenses evolved in different environments and how that shapes their interaction with herbivores. We also focus a lot on functional traits and coexistence and community ecology and how those interact, how those affect interactions between plants and herbivores. But today I'm going to talk about invasion and biocontrols. I'm going to talk about a project that we've been working on with Air Potato over the last 4 or 5 years. So a little outline for my talk. So I'm going to address 3 main questions. The first is, do herbivory rates differ between native and invasive plants? So this is one of the core predictions of one of the most prominent hypotheses in invasion science, the enemy release hypothesis. So I'll talk about differences between native and invasive plants, and then I'll focus more on biocontrol and look at interactions between plant defense and how that affects the performance of biocontrol agents. And then finally, I'll talk a little bit about how multiple biocontrol agents might interact with each other. So being in Florida is a really great place to study invasion. We're #1 in invasive species, or at least close to it. We're in the top 2.5%. We're an invasion hotspot, so we're bright red on this map in terms of invasion hotspots. And what this looks like, if you walk around Florida, you might see pictures like this. So on the left is air potato, just totally dominating the understory of the forest. It's a vining plant, so it even crawls up the stems of trees and gets up into the canopy. And on the right is coral ardesia, another problematic invader that you'll see around Gainesville here with these bright red berries. There's also a fern in this picture too, which I think is invasive as well. So you don't have to go far to find many invasive species in Florida. And these invasive species cause a lot of ecological and economic damage. So every year, the United States is spending almost $500 million controlling these invasive species, and that includes the damage that they cause, economic damage to agricultural systems, forestry, aquatic systems. This is plants and animals, but again, the United States is very high on the list of the damage caused and the amount of money we spend on controlling invasive. So I want to take a step back and think about how these species become invasive in the first place. So we know they're here. We know, that some of them are very problematic and causing ecological and economic damage. but how do species become invasive in the first place? So if we think of a plant in its native range, this is the predictions of the enemy release hypothesis, one of the most prominent hypotheses in invasion science. So this assumes that plants, I'll just focus on plants here, but this could be anything that could become invasive, insects or other animals. So if we think of a plant, it should be suppressed by herbivores or its natural enemies in the native range. So that's keeping its populations in check, keeping it from becoming too dominant and kind of growing out of control. So plants are kept in check by natural enemies in the native range. But if they're introduced to a different area, they might escape those enemies. So now they're released from their enemies. So this is the enemy release part. Things don't necessarily, everything that's non-native isn't necessarily invasive. But if they're released from enemies and they spread and they grow and they dominate ecosystems and become damaging, then we can call it invasive. And this is often what happens if things are released from the natural enemies. So these are the predictions of the enemy release hypothesis. So this was first proposed by Charles Darwin in The Origin of Species. He discussed kind of these general ideas. He didn't really call it the enemy release hypothesis explicitly. But then it was formally proposed by Charles Elton about a century later in this book, The Ecology of Invasions by Animals and Plants. So again, those predictions are just that enemies or herbivores limit population size in the native range, and plants escape herbivores upon introduction to a new area, and then the lack of enemies allows plants to become invasive. So to combat this, we've developed what we call classical biological control, which is just the intentional introduction of those natural enemies into the invasive range that then should suppress those populations and maybe not totally eradicate them, but keep those populations under check and reduce the amount of ecological and or economic damage that they cause. So the enemy release hypothesis is the central hypothesis motivating classical biological control, which again is the definition is the intentional introduction of specialized herbivores or enemies to suppress populations of native species. So the enemy release hypothesis has received a ton of attention in the ecological literature. So this is a meta-analysis from 2004. Already by this time, there was dozens of studies that tested the enemy release hypothesis. So I'm just going to read the first sentence and the last sentence because it kind of sums up the whole paper. So the first sentence is, a recent trend in invasion ecology relates the success of invasive species to reduce control by enemies such as pathogens, parasites, and predators. So the enemy release hypothesis. And then some info on what they actually did. But the conclusion, given the complexity of processes that underlie biological invasions, we argue against a simple relationship between enemy release and the bigger abundance or impact of invasive species. So it definitely happens. There's definitely cases where things are released from natural enemies, but the evidence is mixed, right? So this was in 2004. And that was kind of the conclusion from over 20 years ago. But people are still thinking about this. There's still testing the enemy release hypothesis. There's still a lot of work in this area. It's still, as of 2023, this is a newer paper. They call it the enemy release hypothesis is the best known hypothesis explaining high performance, that is rapid population growth of exotic species. So there's still a lot of interest. There's still a lot of work on the enemy release hypothesis. But what they outline in this article is that previous meta-analyses, not necessarily just that one that I just showed, but previous meta-analyses and other studies might miss a lot of things. They're not necessarily comparable due to differences in environmental context or other factors that might determine whether something might benefit from enemy release under certain circumstances, but maybe not under other certain circumstances. So some of those environmental context things that they talk about are time since introduction. So typically we think of species that are recently introduced. Those should be the ones that are most released from herbivores. And then it might take a little time for the herbivores to be introduced and naturally colonized or introduced intentionally through classical biological control, or native herbivores just adapting and being able to host switch to feed on those plants. So through time, we expect the enemy pressure to build up on this species. So if we compare a study that was done early to a study that's done late, that could contribute to some of this mixed evidence. Or other environmental contexts. So the resource availability or some aspect of the environment in the species home range could determine how defended it is, how many herbivores it has. Same thing in the native, in the introduced range. Things like productivity or the diversity of herbivores could determine the potential for enemy release. Okay, so now to this question, do herbivory rates differ between native and invasive plants? So I'll talk a little bit about this, one of the central predictions of the enemy release hypothesis. So for this, I'm going to use data from the Herbivory Variability Network. This is a network that I'm part of. I'm a PI on this NSF Research Coordination Network. So the goal of this project wasn't necessarily to focus on native versus invasive plants. The goal was to describe how and why patterns of herbivory vary across the tree of life and around the globe. So we have a lot of data from this. And what we did was we designed a standardized protocol and then recruited people from around the world to follow the standardized protocol, measure herbivory on a bunch of plants using the same protocol. So the methods are all the same from all these different surveys. And then we consolidated that, combined it into a massive data set. Yeah, so we have a lot of data on herbivory. So what we have is 127 research teams, and on that first paper, there was about 250 authors that contributed data and wrote the paper. So massive effort. It was a little challenging working with so many people, but it was also great. Creates really good opportunities. So we surveyed in total 519 species from 186 plant families, and we aimed for about 60 plants per population. We also consolidated this into a manual. So if anyone measures herbivory and is interested in following our protocols, this manual is now available online, freely available. There's a lot more than just the protocol, but that is available if you do want to measure herbivory on plants. Okay, so for this study, we had a decent number of native and invasive plants. So we categorized each survey we had as either native or non-native and or invasive. So we had about 504 species that we could categorize, about 355 native, 112 invasive that were documented and classified as problematic invaders, and then about 37 that were naturalized non-native species that didn't necessarily cause ecological damage and weren't classified as invasive. We also gathered environmental context variables, so additional variables related to plant density, so how dense the plant was at the focal site, so whether it's dominating the ecosystem or whether it's relatively rare. Plant diversity, which is shown on that map there, which is related to ecosystem productivity or insect diversity, things like that. So it's kind of a proxy for other variables. And then importantly, time since introduction. So how long since the first record of it being introduced into its native area or non-native area. And when we look across all the species that we have, there's no difference in herbivory rates between native, invasive, and non-native plants. All of them have pretty low herbivory, about 6% damage, and this was pretty consistent regardless of what status it was. So we didn't find any differences. This is across all 504 species that we surveyed. There was no difference in herbivory rates on native, invasive, or non-native plants. For 10 species, we also had surveys that occurred in both the native region and the non-native region. And when we look at these 10 species, there was also no difference between surveys done in the native region or the invasive region. Some of these, we didn't have too many surveys, one or just a handful of surveys, but for 3 of these species, Plantago lanceolata, Plantago major, and Taraxacum officinale, we had at least 20 surveys in each of the regions, so quite a good sample size. No difference even between any of those that were very well sampled. And for Plantago major, we actually found a slight trend for the opposite, where the invasive populations are actually getting a little bit more damage than the native populations. So no difference between native and invasive populations for any of the 10 species that we had surveys for in both regions. We also found no effect of any of the covariates we looked at. This is just a handful of them. We looked at a bunch more, but plant cover, there was no relationship. There was no difference between native and invasive plants. Same thing for plant diversity. And then surprisingly, time since introduction, it didn't matter. This wasn't significant, and it was actually slightly opposite of what we would expect. So earlier time since introduction, so a plant that was introduced only 10 years ago, it got maybe a little bit more damage than things that were introduced over 500 years ago. Okay, so do herbivory rates differ between native and invasive plants? No, we didn't find any evidence for that. Similar damage rates on native and invasive plants. We found no effect of time since introduction or any other covariates that we looked at. So what this suggests is that the enemy release hypothesis is not a good general explanation for how species become invasive. But there are definitely examples. There are cases where this does happen for sure, right? So on average, it's not, very common, but there are still examples when it does occur. So then if it's not occurring very common, it's like, will biocontrol actually work? And biocontrol is still widely used today and for the last century. So these are data from this iBiocontrol catalog. That is a massive catalog of all the introductions across the world of classical biological control agents. For plants, this includes 1,555 intentional releases of biocontrol agents, 468 insect species that have been released targeting 175 plant species in 90 countries across the world. So as you can see, the release of new biocontrol species peaked in the 90s. We're still releasing new species. Old species are still being released as well. So we're still doing biocontrol, but it has tapered off a little bit for sure. So how effective is it? So, if most plants aren't really escaping enemies, how effective is classical biological control? The good news is most establish. So if we introduce them, most of them do establish. But the control or the impact that they have on the invasive plants is highly varied. So this is data, again, summarized from the iBiocontrol catalog. So this is the not established in that kind of light blue. So about a third of insects just don't establish. We introduce them, they don't establish. About a third have no unknown or slight impact. So we introduce them, they establish, but they don't really control the plant at all. And then about a third do have some impact, medium, high, or variable. And variable just means that they can have an impact in some locations, but not in others. So they do have an impact. And this is just anecdotal, but I've been thinking about what are common characteristics of plants and biocontrol agents, plants that might escape enemies and biocontrol agents that might provide successful control. And to me, it seems like a lot of the examples that we have of successful biological control are plants that have interesting defenses. They're very toxic or very well defended, in one way or another. So some of these examples include St. John's wort, which is here in Florida. I've seen it here, but it's more problematic out west. Tropical soda apple, which was all throughout Florida, but it's fairly well controlled by this specialist beetle. So tropical soda apple is in the nightshade family. It has interesting glycoalkaloids. Air potato, which I'll talk about in a minute here, is toxic. It's loaded with toxic saponins. It's controlled by the air potato beetle. And euphorbia, there's a couple species, but these have chemicals as well as sticky latex that provide defense against some herbivores. So this is just totally anecdotal, but it seems like some of the successful cases are ones that have interesting chemistry. So I think about those kind of things a lot. So the next question I'm going to talk about is, do plant defenses mediate performance of biological control agents? Is this something that we can use to predict how effective biocontrol agents will be? So the air potato system is great for this because, as I mentioned, it's all throughout Florida and other parts of the Southeast. It's a very problematic invader. It's native to Asia and parts of Africa. It was introduced back in the 1700s and was in Florida by the 1900s. And it's currently classified as a category 1 invasive, which is the worst category. So it's a very problematic invasive species. It's also chemically defended, like I mentioned, with saponins. And there has been a biocontrol agent released back about 10, 15 years ago in 2011, Liliotherus chenai. It's a leaf-feeding beetle. The larvae and the adults chew on the leaves and feed on the leaves. It's native to Nepal and China. It's very highly specialized on air potato. So it's gone through quarantine, it's gone through host range testing. So basically it doesn't eat anything except this specific species of air potato. And interestingly, it's bright red. So FDAX, DPI, just down the road, disoriented here, think that way. They're mass rearing, they've been mass rearing and releasing the beetles for the last 10 years or so. They've transitioned away from this beetle to another one, which I'll talk about near the end of my talk. But they've released hundreds of thousands of these beetles over the last 10 years. So this is just a graph of the number of beetles released just in Florida over this 4-year period of time. And these are the release sites. So people at FDAX and USDA surveyed these populations, measured the amount of damage done by the beetles at these sites. And this ranges, the scale is a little weird, but white is low and black is high levels of damage. So there's definitely places where the beetle is doing a great job controlling air potato. Even around Gainesville, you can see it, and it's, you know, even though it's light on the scale here. But there's places where it does great, there's places where it doesn't do so good. There's still definitely a lot of air potato on the landscape in Florida. So the control is highly variable. And just to show a little picture of what we see in the field. So this is one site that we visited outside of Miami. There's 2 species in this. The first one in the front is air potato. There's also this other thing in the back that kind of looks like air potato, but this is a different species. But anyways, in the front, you can kind of see these leaves that are totally decimated. So this is what the beetle does when it, you know, it can like almost totally defoliate or skeletonize these leaves. So we see that, but we also see some leaves in the back that are totally untouched by the beetles. So even if the beetles are there, they can, you know, hit the population pretty hard, but there's still, you know, untouched leaves. Those populations could produce bulbils, the reproductive air potatoes. So yeah, if they're there, they can knock it back a little bit, but the plant can still persist. And it has a big underground tuber, so even if it gets totally destroyed, it can resprout the next year from that tuber. So it's a hard plant to control. So the conclusion from this paper, this is just the last sentence of this abstract. So they concluded, however, air potato was still abundant in some areas, which suggests the need for additional beetle releases and or the use of other tactics to achieve management goals. Okay, so we looked at this question from the angle of plant defense. So just can plant defenses explain the performance of the air potato beetle? So this was a project led by Jesslyn Kaur, a PhD student in my lab, now a postdoc in the department here, in collaboration with FDAX and DPI, a bunch of people over there. And Diego Salazar, a chemical ecologist, helped us with the developing our protocol for this episode today. Okay, so the first thing we did was went to a bunch of sites around Florida. Not as many as the original, that paper that I just talked about, that Enrique paper, but we visited 11 sites around Florida, both in the south and even up in the Panhandle. Went to these sites and measured plant cover, damage, and we also collected leaves to quantify the saponin concentration. So part of what we found, this graph shows mean damage, so just the percent damage at the site level. Each point is an individual site that we surveyed. And then on the x-axis is the percent cover of air potato. So what we see here is there's some sites that get a ton of damage, right up to 40%, a lot in the 20% range, but also some sites that get almost no damage. So very high variability in the amount of damage done across these different sites. And unfortunately, the worst sites, the most invaded sites with really heavy air potato cover are the sites that have very little air potato damage by the beetle. And all of these sites had the beetle release, so they were either there or at some point they were there. We saw them at like 9 or 10 of the sites at least. But yeah, so what that suggests is these really problematic populations are just really hard to control. It's hard for the beetle to overcome that, that mass of air potato leaves, or it might take them several years to build up populations and not get back. We also measured saponins in the leaves. So here, this is the same data. So we've measured 5 to 10 leaves per site. So this would be one site here. The point is just an individual plant. And what we found was a little surprising. So the sites with more damage actually had more of those toxic chemicals, more of the saponins in the leaves. So that was interesting. So we wanted to follow up with that trend. And what we would hypothesize is that these saponins, because they're a plant defense, they have some, anti-insecticide, anti-herbivorous properties. We predicted that they should decrease feeding by the beetles, right? If they're plant defense, they should reduce damage by the beetles. So we did an experiment in the greenhouse where we had control plants with no damage. We had unsprayed, but we allowed an insect to feed on them and damage them. We had plants that were sprayed with JA. So JA is jasmonic acid. It's a plant hormone that triggers defense systems when a plant is attacked, usually by chewing herbivores. So plants can upregulate in response to being damaged by chewing insects. So we predicted that beetle damage should decrease on these plants that are better defended. And we also sprayed some with SA, or salicylic acid, which is another plant hormone that triggers defense systems against pathogens and viruses and increasing sucking insects. And typically there's a negative crosstalk between the JA and SA pathway. So if the SA pathway is upregulated and they're well defended against pathogens, they should be more susceptible to chewing herbivores. So we predicted that beetle damage should increase on those plants. And what we found we measured saponins in these plants, and there is some baseline level of saponins, there's some constitutive level of saponin production. So even in the control plants, they're still producing these toxic chemicals, but they do upregulate it. They induce it when damaged or when sprayed with JA. The concentration is about double. And then for chewing damage, we found that they actually preferred the plants sprayed with jasmonic acid. So the induced plants, they seem to prefer those plants. And they almost ate none of the plants sprayed with salicylic acid. So that's interesting. It's opposite of what we predicted, but yeah, so it seems like these saponins may be acting as a feeding stimulant, or there's something about them that they prefer. We also did a 4-way olfactometer with these same plants. So we put them in one plant, each of these treatments, in one arm of this 4-way olfactometer, and measured the time spent that we put a beetle right in the center of that, and then measured the time spent in different arms. Very similar result. So here the beetles are spending more time. The effect size is pretty small, but they're still preferring the beetle damaged plants or the ones sprayed with jasmonic acid. So it does seem like there's something about previous damage or these saponins that the beetles are preferring. So then this led us to this question of, is Lilioceras chennae sequestering? So remember I mentioned it's a bright red beetle, so it's potentially aposomatic, advertising to its predators that it might be toxic. So we asked this question of, is it sequestering? And typically what we expect for the relationship between insect performance and level of toxicity is a negative relationship. So if generalist herbivores are susceptible to toxins, the performance should decrease. For non-sequestering specialists, a lot of times they can tolerate or detoxify chemicals up to a certain point, but eventually are overwhelmed. So they might have this slower slope. But for sequestering specialists, we actually expect the opposite. So they might actually increase performance up to a point and then eventually be overwhelmed by these chemicals. So we tested this using a feeding experiment with artificial diet, where we mixed diosgenin, saponin compounds, into artificial diet at concentrations that match what we see in the field, plus a little beyond. So it was beyond the range that we naturally see in the field to get those extremely high concentrations. We put adult beetles on the plants with 24 replicates per treatment. And we allowed the beetles to feed for 14 days. And we measured the frass that they produced, and a couple other things, but I'm going to show frass mass here as a measure of how much they're consuming over that 14-day period. And what we see is this hump-shaped relationship. So they're doing best, they're eating the most at the intermediate concentrations of diastereonin. So at the extremes, when we added 0 or when we added the really high amounts, they did the worst. They decreased their feeding. We also looked at survival over that 14-day period, and the same pattern. So they survived overall really well during this 14 days, but the highest mortality was at 0 and 20 milligrams per gram of diastereonin. So overall, this does support what we would expect for a sequestering specialist. We haven't actually ground up the beetles and looked for the saponins in the beetle bodies yet, but we're hoping to do that in the future. So do plant defenses mediate performance of biocontrol agents? So yes, we found that saponins actually improve the performance, the amount that they feed, as well as the attraction to the plant. And Xavier Martini has done some similar work on the volatiles released, not on the saponins, but it seems like they can hone in on these volatiles that are released from chewed plants. And this may translate to improved biocontrol in the field. So if beetles are able to find new populations based on where other beetles are, that's good. It might help them find new populations or decrease the numbers populations that they are currently at. And I think this has broad applicability, both the specific things we found with saponins, but just more generally looking at interactions between plant defense and the effectiveness of biocontrol agents, because all biocontrol agents that are released are specialist herbivores. They have to go through a quarantine procedure and a host range testing. So we know that they're basically specialized on their specific host. So I think there's a lot more interesting things that could be looked at more broadly here. Okay, so now I'm going to shift to the last topic. Are there synergies among biocontrol agents? So far I just talked about that one beetle species, but if this is again data from that iBiocontrol catalog. So on average, most invasive plants that have a biocontrol release have many release for them, about 3.25 on average. And this ranges from 1 to 44. There's one plant species that has 44 approved biocontrol agents released for it. It's lantana, which we can see in Florida here. It's pretty crazy. And some of them work, most of them don't, but most plant species have somewhere in the range of 1 to 5 or 6. Once we get up to 10, it becomes pretty rare. So when we think about how multiple biocontrol agents might interact on the same host plant, there's several hypotheses or several models for how they could interact. One is the cumulative stress hypothesis. This is kind of the default expectation. If we introduce one biocontrol, we kind of hope that other ones would add cumulative stress. So this is kind of the general expectation. And it's just that multiple agents are necessary to overcome the invader's reproductive potential. So if we think of one beetle that might be attacking the leaves and some grub or something that's attacking the roots, the combined effect of these two herbivores might be needed to overcome the plant's ability to grow and reproduce. There's also the lottery model. So this just predicts that if you increase the number of biocontrol agents that you introduce, there should be a higher probability that one of those is super effective at controlling the plant. So kind of the silver bullet, you know, if you throw 10 herbivores out there, one of them might have a good chance at controlling the plant. So we might introduce a caterpillar that maybe doesn't establish, you know, we introduce some beetle or something that maybe has weak effects, and then you get lucky on the third one that really provides efficient biocontrol. Or there's another one, the synergy model, which is similar to the cumulative stress, but it predicts that there's a facilitative effect. So one herbivore is necessary to change the stress of the plant or the chemical profile or the nutrition or something of the plant, some aspect of the plant that is necessary for the second herbivore to then come in and provide effective control. So air potato is another convenient system for this question because there's a second biocontrol beetle that was released, and Rosie took these pictures here. So a couple of years ago, Lilioceras eugenia was approved and started to be released on the landscape. So it's the same genus as Lilioceras cheni. Looks almost identical. They're very difficult to distinguish. They're both bright red. But one feeds on the leaves. Cheni feeds on the leaves. Eugenia feeds primarily on the bulbils, the aerial reproductive tubers. So this is a bunch of them swarming the bulbil. The adults feed on the outside, the skin, and the tissue as well. They lay eggs on the inside of the bulbil, and then the larvae develop inside the bulbil, kind of eat it from the inside. So we think that there might be synergies between these two biocontrol agents. Just to remind you, L. cheni prefers high saponin levels in the plants and plants that are previously damaged. So if these plants are inducing systemically and inducing those higher saponins in the leaves as well as the bulbils, and if Lilioceras eugenia has a similar feeding pattern, we think there could be a synergy between attack first on the leaves by cheni that then increase feeding by the bulbil feeder. Yeah, so we just asked, will it prefer bulbils that were from plants that were previously damaged by L. cheni? So this is more work by Jesslyn Carr for her dissertation, as well as Lucia Navia did a lot of work in the lab, collecting a lot of grass and doing these experiments, as well as with folks at Dex. So for this, we used the same 4 treatments that we used in that previous experiment. So we grew plants that had no damage, just control plants, plants that were unsprayed with any hormone but damaged by the beetle, or sprayed with jasmonic acid or sprayed with salicylic acid. We let them grow for about 3 months to produce these dumpling-looking bulbils, and then we harvested those bulbils and fed them to the larva of Lilioceras cheni in these little cups. So we put freshly hatched first instar larvae in these cups and let them feed until they pupated, which takes about 10 to 14 days or so. And collected frass every day as a measure of feeding. We also measured pupil weight and bulbul damage at the end. And for the bulbul damage, we actually used beeswax with this protocol developed by Alan Dray and Goldstein. But the larvae create these feeding tunnels in the bulbils, so you can fill them with wax to get like a volume of how much they would consume. So we did those measurements. And here again, I'm just showing frass weight because it was very highly correlated with the bulbul, the area consumed. But no difference between these treatments. So no effect of beetle feeding or any of the plant hormones, maybe a little bit more feeding on the JA or SA ones, but no significant difference here. But then when we looked at pupil weight, the pupa were a little bit heavier, a little larger on the bulbils from plants sprayed with salicylic acid, which again isn't exactly what we expected. Cheni preferred the beetle or JA plants, and didn't like the SA plants at all. But here, these beetles are preferring bulbils from the salicylic sprayed plants. So again, we wanted to look at, are they sequestering? We expected that they probably would because they're very closely related to cheni, which we found this pattern with cheni. But because of the differences in what I just showed with what bulbils they prefer from different plants, maybe we might see something different. So we did the same experiment before with this artificial diet and added diasgenin at different concentrations and found a totally different pattern from what we found from cheni. So here we found that they're decreasing in a dose-dependent way. So the more diasgenin, the worse they actually did. So are they sequestering? Probably not. And it could be because these bulbils are just so starchy that like the relative concentration of saponins to starches just makes them less susceptible to the specific chemicals. And they can, they're just feasting on these, great starchy masses. But yeah, totally different feeding patterns between these two, otherwise, almost identical species. So are there synergies among biocontrol agents? So not exactly synergies, but we did find support for cumulative stress. They're not really affecting each other in their feeding or how they affect the plant, but rather one is feeding on the leaves and damaging the leaves, the other is feeding on the bulbils, damaging the bulbils, but they're not affecting the plant in a way that negatively or positively affects the other species very strongly. So my new student, Zane Holditch, is following up on some of these questions because we haven't yet done like damaging the bulbils and seeing how that affects Liliaceus chenna, the leaf feeder. So he just started in the fall, but he's already done some pilot experiments. So these are similar treatments. So here he damaged them with Eugenia, the bulbils a little bit, and they can still, if they're not fully damaged, they can still sprout. So I think he had 10% or so, of the bulbul eaten, and then planted it and grew a plant from these damaged bulbils. And then either sprayed them with JA or no JA. And it was kind of a small pilot experiment. So no strong significant effects, but there was a trend towards the leaf feeders preferring the plant sprayed with JA. So very similar to what we found in our previous experiments, but no effect of the bulbul feeding damage. So it seems like the damage on the bulbul doesn't really affect the plant quality or the subsequent feeding by these leaf tubers. And then just one interesting thing. So he also looked at the sex ratio for Eugenia, and it seems like females are doing a lot more damage than males. They're similar size, but they have to support eggs and produce eggs. So it seems like they are doing the most damage. So this is just the sex ratio and a slight increase in feeding with more females doing the damage. Okay, so in general, it does seem like two beetles are better than one. One's hitting the bulbils, one's hitting the leaves. So their combined effect should be additive at least, but if you have one, that's great. If you have two, it's even better. Okay, so are there synergies among biocontrol agents? Not synergies, but rather we found support for the cumulative stress hypothesis. But one important thing is we haven't yet done these experiments with both beetles on the plants at the same time. We've looked only at indirect effects and how they change the chemistry of the plant, but they could interact antagonistically if they're both on the plant at the same time. So that's kind of what's planned in the future. Okay, so some quick takeaways. So enemy release hypothesis, we found no support for it really as a general explanation for how species become invasive, but plant defenses do seem to be important for determining the performance of biocontrol agents that are released and do have some success in the field. And no synergies, but it does seem like we haven't found any negative interactions between these beetles. They're not competing, they're not interfering with each other as far as we know so far. Yeah, supporting cumulative stress. So the next steps is to do these field experiments, do stuff in the field with both beetles on the plants at the same time, which, yeah, might reveal new things on how they interact. Okay, with that, just a bunch of acknowledgements. Everyone from my lab in the last few years, everyone at least contributed to the air potato stuff in one way or another. And then colleagues at FDAX and DPI have been super helpful with their mass rearing program. We can just go over there anytime, get beetles. We've done experiments in greenhouses there, so FDAX has been great. Other colleagues at other institutions and funding sources. And with that, I'll take any questions. Yeah, really.
For your presentation, I just had a question about the first study you talked about with the herbivory variability network. How did you measure plant?
We just used a quadrat and did percent cover of the focal species. We also counted the number of stems, but this was the percent cover method that we had.
I noticed on your map that you only had a yellow spot on Florida for native species.
Good question.
Well, the question, I mean, and then you talked about some situations where the enemy release hypothesis does hold up.
Yeah,
And I'm thinking specifically about coral rhodesia, which is to my knowledge, nothing eats it.
yeah.
Is that one of the exceptions?
Well, yeah, I don't know about, I think the chemistry is really interesting for that. It's in the coffee family, I think, so it probably has interesting leaf chemistry. I don't know if they've, yeah, there's no biocontrol agents released for it, but that could be the case if there's something, especially that damages like the fruit or the berries, that could be really effective. But it'd have to be a highly specialized thing. So it does have those characteristics where it's, probably very toxic. No native herbivores eat it, but it could be susceptible to highly specialized herbivores. I don't think that, yeah, they haven't discovered anything yet, or at least, I don't know if anyone's working on anything in quarantine for that. But yeah, it fits the other characteristics of yeah, those anecdotes.
Right, great talk. I have a couple of questions, or ones are sort of the very brief ones.
Yeah.
The bulbous feeding ones, do they feed in the ground or in the air?
Good question. We don't actually know a lot about them in the field, mostly on the ground. The only things I've seen are on the ground. I've never seen them on a bulb on the plant. And then we have some in the greenhouse, and if we put them on a plant with a bulb on it, they eat the leaves. Usually they don't eat the leaves, but it seems like they prefer leaves as opposed to bulbils on the plant. So usually it's on the ground.
So I guess to follow up with the kind of two parts. The first is, if they're feeding on the bulb, did you check the changes to the sapling levels in the leaves? And then the immediate follow-up is kind of more broad, which is when you said there are these exceptions to the rules. What do you hypothesize are what drives that to the exception to the rule? Is it the plant chemistry and defense?
Yeah, good question. Yeah, that's what I suspect, is that there's certain characteristics, like a really defended plant with a really tight linkage with a highly specialized herbivore. Those are the ones that seem to be successful. It's interesting. It would be interesting to look more at that big catalog. It's really hard to compare different species and how defended they are, because chemistry is just crazy. Like there's different classes and it's hard to compare them in any way. Same thing with herbivores. Like it's hard to get a list of all the specialized herbivores in a plant species. But yeah, that's what I suspect, is that that tight linkage between a highly specialized herbivore and a plant that is otherwise protected, from native herbivores and other generalists. Those are the cases where biocontrol may be most effective. Yeah, wait.
So in the field, like in the natural world where these beetles are being released, and let's just take this from a single plant perspective. So when the plant has been invaded by the foliar eating beetle, is it safe to assume that it produces fewer tubers?
Yeah, so that has been shown. Yeah, so the more damage, it'll produce less of those bulbils.
And so as far as you've observed so far, the tuber feeding or the bulbul feeding beetles are invading from the soil, not while it's hanging on the plant.
Yeah.
So is it safe to assume that whenever a bulbul has been invaded, that it will not produce a new vine?
Usually if it's Zane can probably answer this better, but if it's the adult beetles feeding on it, they don't do a lot of damage. They chew a little bit on the skin and they chew into the bulbul a little bit, and then they lay their eggs in there. Once there's a clutch of eggs in the bulbul, it's usually totally destroyed. They'll just totally empty out the whole bulbul, or they'll eat it enough that it just rots from there.
So from a beetle reproduction perspective, you're not seeing any synergy, but from a plant reproduction perspective, you're seeing synergy. You're seeing a synergistic effect with the effectiveness of control. So with the two beetles in tandem, you're really knocking its reproductive potential.
Yeah, exactly. So yeah, it's like an additive effect. But this is stuff we haven't tested in the field yet. We don't really know the phenology of the two beetles. The leaves aren't, they die back in October, and then they sprout again in April or May. And in the meantime, there's some bulbils on the ground. But then there's a period where there's only leaves, and then they produce new bulbils in spring or at the fall, in the fall. So we don't know what the bulbul feeder is doing when there's only leaves. In the winter, the other one goes into diapause or pseudo-diapause, and then overwinter is just, kind of quiescent, doesn't really eat that much. But the bulbul feeder seems like it's active throughout the winter, eating bulbils. So those phenological questions, we don't really know. But so they might interact negatively if if there's only leaves and they're both on a plant and there's no bubbles. Yeah, we just don't know that much about the natural phenomenon.
And real quick, can you pull up the slide that shows your beetle damage with the release sites?
Yeah,
That map that goes from the Louisiana, the Mississippi Delta, all the way to South Florida?
yeah.
Because, well, maybe I can, maybe I can ask you later,
Yeah,
but like, I just at a glance, I didn't get to look at that slide very much, but at a glance, it looked like there was. Like, there could be a physiographic, factor going on,
yeah, like it was most in the Panhandle in South Florida.
or like in the swamp of the Delta and in the Everglades.
Yeah, true. Yeah, interesting. Yeah, so there was a survey, a different paper in 2016 that had kind of like totally inverted damage patterns. Like it was highest in Central Florida and really low in South Florida
Huh.
and the Panhandle. Yeah, so I don't know why that is, but it could just be the beetle wasn't yet spreading to those extremes. In the Panhandle, and I guess the Panhandle is still low, but yeah, there's a lot of interesting phenology questions that I don't quite know yet. Rosie.
I was also wondering with this map, so like I know this is like a comprehensive survey over several years, but like air potato used to be a really big problem in Gainesville and it's not really anymore. So like, does this damage, is that taking into account the amount of air potato in the area? Cause like, there's not gonna be a lot of beetle damage in Gainesville because there's just less air
Yeah,
potato
sure.
in Gainesville now.
Yeah, yeah, that's a good point. Yeah, because there's sites all around Gainesville that are low, and you would like every place I go, I see the beetle. Yeah, Eric.
I was just gonna say, so I was on those studies on the papers, we randomly, within each county, we wanted to find 2 sites randomly, but some made, especially in the panhandle only had 2 sites. So, and exactly what Rosie said, the releases, we started in Florida and then we started spreading out west, so there's a time differential between them of years. And it's the scarcity of the actual vine. So in the panhandle, it was very scarce in areas. And there was areas we couldn't get to because they didn't want us there for whatever reason. And so there are other factors in this, but it was also a snapshot in time. So we just went around one season, we hit all the counties. So there's also a difference seasonally going up and across. So we couldn't hit all of, the vines in every area within the same growth period, if that makes sense.
Yeah, damage probably builds up throughout the season.
Because it just took forever.
Yeah.
So we might have started when vines were really sprouting, but then towards the end, some were not even sprouting anymore.
Yeah.
So it's just a snapshot, really.
Hmm.
It's not a comprehensive review of the effect.
Yeah.
Sorry, I didn't mean to say all that.
No, that was, yeah, that's.
This one.
I had a question about the first study, because I was surprised about that. And I'm wondering, like, how you account for what you don't see. And by that, I mean, presumably you're surveying where the plants are.
Yeah.
Could it be that native plants do have more damage and that is just resulting in fewer populations, fewer individuals? So essentially the absence of plants.
Yeah.
How do you account for that? Does that make sense?
Sort of, yes, it does. It's hard to account for. So yeah, I mean, it's not perfect. Like, to get at that question, I think you would have to do at least multiple years of surveying. Ideally also with like an insecticide treatment where you exclude herbivores.
Yeah,
This is all observational data, so
Right.
it's quite limited in that sense. But yeah, so there's the absence, like
Yeah.
they could just be,
Yeah.
excluded from some sites. Same thing with the amount of damage, like if a plant is totally eaten, we wouldn't,
Right.
we wouldn't
Right.
measure it. So yeah, I mean, there's definitely limitations to those big surveys.
Yeah, it's almost like the fact that the plant is invasive. You almost have to include, yes, enemy release is happening here, right? And
Well, I mean, there's other explanations for how plants are invasive. There's like a 20, 30 hypotheses. It could, could be open niche or propagular pressure. The enemy release, that's the only one I talked about, and it's one of the more well-known. But, I mean, things can become invasive, totally unrelated to enemies. It could be soil conditions or resource multitudes. Yeah, so it seems like enemy release happens, under very certain circumstances. But most, yeah, so it's probably a mix of it, it could be partially enemy release, but partially tropical pressure where they're just continuously coming in constantly, leading to their invasion. So yeah, it's probably a whole host of policies.
I think we had the last question from the river, but can read and.
Are there any differences in the number of different herbivores in the native and invasive range on the plant? Do we know anything about below-ground pests and pathogens of these plants? Great questions. For air potato, they've done a bunch of surveys in the native range. So there was a list of a bunch of potential candidates for biocontrol. And then the air potato beetle emerged as the one that seemed to be most effective. But yeah, there is a handful of, at least for air potato, there's a handful of other natural enemies in the native range. Or just in general, for the stuff we did across the globe, it's really hard, like a lot of times you see the damage, but you never see the herbivore that caused the damage. So we have some data on the insects that are present, but it's very sparse and kind of unreliable. That's another thing that would involve, like you'd have to do more intensive sampling. Below ground stuff, I don't know anything about that. It's a lot harder to do. We haven't looked at that yet. So that's another limitation. Like we've only, looked at chewing damage. We're starting to measure damage in reproductive structures, but we haven't done anything below. But so there's an opportunity there, Jordan.